Okay, wonderful. So we are very pleased to have today with us in our seminar a very special guest from the West Coast, Professor Kalanit Grill-Spector from Stanford University. Kalanit is a professor of psychology and the Wu Tsai Neurosciences Institute at Stanford. She studied electrical engineering and computer science at the Ben-Gurion University of the Negev, and then did a PhD at Weizmann Institute of Science with Professor Rafi Malach, where she started to investigate the human visual system using fMRI tools to reveal exciting results about the human visual cortex as cue invariance, the characteristics of the lateral occipital complex and its partitions as revealed by fMRI adaptation, a method that she developed together with Rafi. She then continued to do a postdoc at MIT together with Nancy Kanwisher, and after that she joined Stanford as faculty and opened her Vision and Perception Neuroscience Lab. Throughout her career, she has published many influential papers in top leading journals as Science, Neuron, Trends in Cognitive Neuroscience, Acta Psychologia, and many others. Her work is highly cited. She has an H index of 51 according to Google Scholar. She has supervised many successful graduate and postdoc students, some of which have already continued to successful careers of their own. She won multiple prestigious fellowships as the Human Sciences Frontier Fellowship, the Sloan Fellowship, the Klingenstein Fellowship in Neuroscience. She has also served as editor for the Journal of Vision and Neuropsychologia journals. On a personal note, I want to thank Kalanit for exposing me to the field of cognitive visual neuroscience and being my instructor and inspiration when I decided to join the field and started my way in it. Kalanit investigates many aspects of human vision and includes both functional investigations and anatomical ones. She investigates them both from a systems level approach, as with fMRI and connectivity of the structure, fiber-wise and tissue-wise, and also at the cellular level with causal stimulation studies in the human brain to reveal exciting findings about the visual system. She also uses computational methods, deep nets, population receptive field mapping, and elaborates into developmental studies looking into how the human visual system develops. So without further ado, we're very pleased to have you today with us, Kalanit. Thanks so much for joining us today to tell us about your research.
So thank you, Sharon, for this very kind introduction. It's really great to be here in Israel in Ben-Gurion, Bar-Ilan University, or virtual Bar-Ilan University. And it's really great to be able to reconnect with old friends. So I'm very excited to be here. So let me share the screen. And Sharon is going to monitor the chat. So if you have questions, she's going to relay them to me. Let's do it. Okay. The screen okay, Sharon?
Yeah, wonderful.
Okay. So what I'd like to tell you today is a work that's been ongoing in my lab for many years, and it's using the human visual system as a model into the developing brain. And this work is very labor intense. I'm going to tell you today about developmental studies that we collected data over a period of 5 years. So a lot of students and postdocs were involved, and these are all the students that participated in this research. And in particular, I'd like to highlight the contribution of Marissa Nord, who's on the left, who is the first author of many of the studies that I'm going to show today. So recognizing faces and reading words is important for everyday lives. However, our ability to read and recognize faces improves over childhood development. For example, 17-year-olds are better in reading than 5-year-olds, and 17-year-olds are also better than 5-year-olds in recognizing faces, whereas there are adult faces or children's faces. And the question I'm going to address today is, how does visual cortex develop during childhood to enable improved visual recognition abilities? And in particular, I'm going to talk about the intersection between brain function, brain anatomy, and behavior, or cognitive neuroanatomy, as was given a name by Kevin Weiner. So to study visual recognition, we study the ventral visual pathway. So in brief, as light gets reflected from objects in the world and images formed on your retina, This information is then relayed through the optical tract to the occipital cortex, where it hits primary visual cortex, or V1. And then through a series of visual areas or processing stages, it reaches the ventral part of temporal cortex, this area shown here in yellow, called VTC, and a percept emerges. And we are interested in studying ventral temporal cortex is because it has both distributed and cluster representation that seem to be useful for object recognition. For example, if you look at the distributed pattern of activity shown on the left, you'll find unique patterns of activity for each visual category, and this allows fast readout as well as representation of multiple categories. In addition, within this stretch of cortex, there are these regions shown here in color that seem to be specialized for processing information for particularly ecologically relevant categories. For example, on the fusiform gyrus, you'll find bilaterally 2 regions that are selective to faces. On this occipital temporal sulcus, this region shown here in orange is a region that is selective for limbs. Also on the occipital temporal sulcus, and more prominently in the left hemisphere, there are regions that are selective for words. And in this collateral sulcus, this region shown here in green is selective for places. And what do I mean by selective is that if you measure responses, let's say within the face selective regions to many different kinds of faces, you'll find that the amplitude to many individual faces is really high. But if you compare this to the amplitude of response to other items such as limbs or cars or houses, the amplitude is consistently lower. The selectivity to faces is also maintained across formats. So you can see selectivity for faces over objects, whether you present them in grayscale or as two-tone silhouettes. And even when you control for low-level features, for example, if you show an upright silhouette that's perceived as a face, the response is higher when you show the same silhouette upside down, and it's no longer perceived as a face. Furthermore, some research from our group as well as others has showed that these regions are causally involved in the perception of the relevant categories. For example, if you stimulate the face selective regions, you'll find specific impairment in the perception of faces. It is, in the case of the developmental research, these category selective regions are interesting because they can be identified in individual people and even in our youngest children. So what I'm showing you in this slide are 4 left hemispheres of individual people. And you can see that in all of these participants, you'll find the place selective region in yellow, the face selective regions in red, and the word selective region in black. And they also have the same spatial arrangement across cortex. So over the last 15 years, a large body of research has compared responses in these category selective regions across children and adults. And these regions have found that not all of these regions develop at the same rate. And in particular, face and word selective regions seem to be less developed in children compared to adult. But by age maybe 7 or maybe by teenage years, some other category selective regions like object and body part selective regions seem to be similar across children and adults. And this has led, so one of the things that is really interesting is that word selective regions, such as the ones that are in black, develop when children start to read. So this has really led to 2 hypotheses about how these word selective representations might develop in school-age children. So one idea is that cortex goes some refinement with visual experience, and maybe some category selective regions are developed by early childhood, but when you learn to read, not all of the cortex is developed, and some territories that isn't fully developed, shown here in white, will become word selective as you learn how to read. And evidence for this comes from a study that's published several years ago that shows that this word selective regions emerges during the first year of school as 5- and 6-year-olds learn how to read. Another, and this is called the refinement hypothesis. Another hypothesis is this competition hypothesis, and specifically, this hypothesis suggests that because faces and words require the fine visual acuity afforded by foveal vision, they are fighting over a cortical territory that represents a fovea. And maybe young children have already developed some selectivity to faces, but as children learn how to read, this word selective activation takes over some of the face selective activation, and more so in the left hemisphere because language is left lateralized. And evidence for this idea comes from some work, for example, from Marlene Berman and colleagues, that shows that as the lateralization to words increases in the left, lateralization to faces increases in the right hemisphere. Now, addressing these hypotheses cannot be done with cross-sectional studies and really requires longitudinal research within the same child to see how their brain develops. So what I'm going to show you today is some new longitudinal data from our lab that is going to test these predictions of these hypotheses on the longitudinal development of category selectivity. I'm going to start with describing the development of these clustered regions. Then I'm going to talk a little bit about the distributed representation development and how it might relate to development of behavior. And in the end, I'm going to make a link between this functional development and anatomical development. Okay, so to test these hypotheses, as I said, we obtain longitudinal fMRI data in school-age children. Our participants were initially between 5 and 12-year-olds, and we collected data within the same participants over a period of up to 5 years. During the experiment, during fMRI, they looked at images from a large range of ecologically relevant categories. So we have 10 categories of stimuli. Stimuli from each category were presented in 4-second trials. In each trial, they see 8 different stimuli. And infrequently, an image with just a background appears, and they're asked to fixate and press a button on this oddball stimulus. So the children see many different trials like this over 3 runs with different stimuli, and overall they see 144 images from each of these categories. What's really important about our research is that we track development within an individual child's brain. So all the data I'm going to show you are within individual children. We did a lot of quality controls to ensure that we get high quality data in the children. So we only include data that the children don't move and perform really well on this task. So altogether, I'm going to show you data from 29 children and 128 functional sessions. Okay, I'm going to show you today data from the lateral VTC, which is this region shown here in green, because this is a region in cortex where you're going to find face and word selective representations in participants. Data from the medial VTC is in our paper, so if this interests you, you can check the paper. So let's first consider the prediction of these hypotheses on the development of category selectivity. So the first thing that we did is we measured how many voxels were selective to each of these 10 categories. So if we consider the refinement hypothesis suggests that as you become more proficient in face and word recognition, your cortex becomes more selective to these categories. So we should see developmental increases in the number of word and face selective voxels within lateral VTC. In contrast, the competition hypothesis suggests that as children learn how to read, word selectivity will indeed increase, but especially in the left hemisphere, face selectivity will decrease. So to test the prediction of these hypotheses, we measured in each participant the volume of category selective activation. So this is a scatter plot showing data from 128 sessions. Each session is a dot, and the sessions are colored by participant. And the x-axis is age, and the y-axis is the volume of word selective activation in the left hemisphere. And you can see that there is a significant increase in word selective activation from age 5 to 17. Okay, so what about faces? If we measure as the amount of face selective activation, whether when children were looking at child faces or adult faces, we also see a small but significant increase in the amount of face selective activation from age 5 to 17. So this seems to be consistent with the predictions of the refinement hypothesis. But what about the other 8 categories? So to our surprise, we found that at the same time that word and face selective activity increases in the same children, limb selective activation decreases. So basically, young children like 5 to 9-year-old have more limb selective voxels than 17-year-olds. So we can summarize this development by fitting a linear mixed model across these data. And what we're interested in the slope of this model, because it tells us about the rate of change in the volume over time. So now I'm going to show you this slope of this linear model for each of the 10 categories in both hemispheres. So if we look at the word selective activation, if we look at the left hemisphere, we see a significant increase in word selectivity. However, this doesn't extend to the right hemisphere, so it's specific to the left hemisphere. And it interestingly doesn't extend to another category of text, such as numbers. We see this decrease in limb selectivity in both the right and left hemisphere, but it doesn't extend to body part selective activation. So it seems to be specific to limbs. With respect to faces, we find increases in the volume of face selectivity, both in the right and left hemisphere, and for both children's faces, both children and adult faces. And we don't really see development of other category selectivity, except for a small but significant increase in the amount of house selective voxels in the left hemisphere. So one of the things that we can do is that because we're looking at the same child, we can see how this cortex changes over development. So this is an example hemisphere where we're zooming on this word selective activation of this particular child in the initial session. And you can measure in the same child how this word selective activation looks like in the last session. And you can see that this black contour shows that this region grows. But what's really interesting to study is what is happening in this region, what we call this emerging word selective region that was initially not word selective and became word selective over development. So one of the things that we did is that we measured the activity or the level of response to each of the 10 categories from this region in the young children, the 5 to 9-year-olds, and compared them to the teenagers. So you can see that this region is initially not word selective because it responds similarly to words, numbers, and limbs. So the question is, what happens across development? Why does it gain word selectivity? One possibility is that the responses to the irrelevant stimuli, such as limbs and numbers, might decrease across development. However, another alternative is that the responses to words increase across development. So we can examine these possibilities by comparing the 5 to 9-year-old data to the teenagers' data. So the first thing that we found is that the word amplitude of response to words significantly increase. And we didn't actually find any significant decreases to numbers or words or to the other categories. So it seems that this region is gaining word selectivity because there is a specific increase in the responsiveness to words in this region. Another thing that we could look is what happened in this region that loses its limb selectivity. So this is a different child. The yellow region shows its limb selectivity in the first session, and the black region shows its limb selectivity in the last session. And you can see that this region has shrunk. So here we can look at this waning region shown here in red, which is a region that was initially limb selective, but then lost its limb selectivity. So again, we can compare the responses in the young children compared to the teens. So as expected, in the young children, this region is limb selective because it shows the highest responses to limbs. So why does it lose limb selectivity? Do the responses to limb go down or responses to other stimuli go up? So you can see that the responses in teens are lower in the same region. And at the same time, we see trends for increases in responses to words and faces in this region. So we can see that a lot of the changes are happening because of decreases or increases response to the preferred category. However, if we really want to examine the prediction of the developmental hypothesis, we might want to track the changes in selectivity within individual children and not at the level of the group response. So another thing that we wanted to see is what is this relationship between word selectivity, shown here in the vertical axis, as a function of face and limb selectivity in this region that is going to become word selective. So if we consider the prediction of the competition hypothesis, it makes a very specific prediction. This hypothesis predicts that initially this region should be face selective, so the initial activity should be in this quadrant, and as it loses face selectivity, it should gain word selectivity. So if we're going to look at this relationship between selectivity to these three categories at the first session, which is the bottom of the arrow, compared to the last session, which is the top of the arrow, the competition hypothesis suggests that individual children's trajectory should look like the arrow shown on the right. Starts with positive face selectivity and low word selectivity, and it will lose face selectivity and gain word selectivity. So how does the data look like? So this is our data. Each arrow is an individual child. And again, the bottom of the arrow is the selectivity in the first session, and the top of the arrow is the selectivity at the last session. And you can see that the data doesn't really look at all like the prediction, first of all, because it starts in the wrong quadrant. So instead of being, instead of being face selective, it's actually shows negative selectivity to faces. So it's true that it gains word selectivity as we expect, but it actually does not start from being face selective. So to better look at the data, I'm going to flip the axes of this plot, and I'm going to put this limb selectivity in front. So this is the same data, but now I'm putting the limb selectivity in the front and face selectivity on the side. And what you can see is that in all children, this region starts with initial limb selectivity because it starts in this positive quadrant. And as it loses limb selectivity, it gains word selectivity. And we can find this pattern of what we call cortical recycling in 27 out of 29 of our participants. So we have new evidence that limb selectivity is recycled into word selectivity as children learn how to read. We can do the same analysis in this emerging face selective regions, but here we're going to look at the relationship between face selectivity, limb selectivity in the front, and word selectivity on the right. And you can see in this region that is going to become face selective during childhood development, we also see this recycling of initial limb selectivity into face selectivity. And again, this happens in the majority of our participants. So basically what I've shown you so far is new data that shows that during childhood development, there are both increases and decreases in category selectivity. And we have this evidence that limb selectivity that's prominent in young children is recycled into word and face selectivity in ventral temporal cortex. So given this finding of recycling, how does this cortical recycling affect the broader distributed patterns across ventral temporal cortex? And does it have any impact on recognition of these categories? So in the second part, we're going to take the same data, but instead of looking at the clustered activation, we're going to look at the longitudinal development of these distributed patterns. And what we're going to do is we're going to look at the representational structure in children's brain using representational similarity matrix. So this is an example of a distributed response to in one child's brain as he sees one set of words. And you can measure the distributed response in the same participant when he sees a different sets of words. And what we're interested is to examine how similar these responses are. And we do that by measuring the correlation between these patterns, because if these patterns are similar, it would suggest that the representation generalizes across items of a category. And you can see that these patterns are indeed positively correlated. We can do this also for another category. For example, the top shows the distributed response to one set of child faces, and the bottom shows a distributed response to another set of child faces. And again, you can see that there's high similarity or high correlation between the patterns. Another thing that's interesting is how different are the patterns between categories. So if we measure the correlation between the patterns of response to words and faces, you can see that the patterns are not correlated. In fact, they're slightly negatively correlated. And of course, we're going to do this across all pairwise comparisons across all 10 categories. And what you get is this representational similarity matrix. So what you can see on the right is a representational similarity matrix average across 5 to 9-year-olds. And what's interesting that already in this young age, this matrix has a categorical structure because the patterns of response seems to be similar across items of a category and perhaps a domain, and seem to be dissimilar across domains. Another way to visualize the data is to do multidimensional scaling and look at the axis that has the greatest variation of the patterns. And you can see that some categories are well separated in children, for example, faces and maybe bodies, but other categories are not well separated. For example, words and objects are not well separated, and objects and houses are also not well separated. So we wanted to quantify how these distributed patterns develop between age 5 to 17. So to do that, we're going to use a metric that we call distinctiveness. And basically, distinctiveness is the difference between the within category similarity minus the between category similarity. So basically, this index goes between 0 to 2, and the higher this index is, it means that the patterns are more similar across different items of a category and more unique or more different across categories. So what we're going to do is we calculate the RSM in each of our sessions. And for each of our 10 categories, we're going to measure distinctiveness. And what we're interested in to quantify if this distinctiveness increases or decreases as a function of age. So before I show you the data, let's see what could be possible outcomes. One possible outcomes is that distinctiveness would increase with age, because as these selectivity is increased, these category representations become more enhanced, and perhaps we should find increases in distinctiveness for both faces and words that gain category selectivity. Another possibility that's triggered by our finding of recycling is that maybe we should see increases in distinctiveness for categories that gain selectivity, like words, but we should find decreases in distinctiveness of distributed patterns for other categories, such as limbs. Okay, so we're going to measure distinctiveness as a function of age. And I'm showing you data now from the right hemisphere. And this is distinctiveness for child faces. Again, each dot is a session, and the sessions are colored by participant. And we can see a positive increase in distinctiveness, meaning the distinctiveness increases from age 5 to age 17. So what about limbs? So for limbs, we find that distinctiveness actually decreases from age 5 to 17, which is consistent with our recycling prediction. And again, we fit a linear mix model to quantify this development. And what we're interested in is the slope of the model. And we can see that there is a significant and positive slope or increase in distinctiveness for faces and a decrease in distinctiveness for limbs. Okay, so this is when we look at the overall patterns, but lateral VTC has a lot of voxels, and we're curious to see which voxels contribute to this development. So what we did, we separated the VTC into two kinds of voxels. One kind of voxels were voxels that were selective to any of the 10 categories. So we call them the union of the selective voxels. And this constitutes about 35% of the lateral VTC voxels. And the remaining or majority of the lateral VTC voxels are voxels that respond to these stimuli, but don't show preference to any of these 10 categories. So we're going to measure separately the change in distinctiveness over development for the selective and non-selective voxels of VTC. So when we look at the selective voxels, for example, in right VTC, we see indeed increases in distinctiveness for faces and decreases in distinctiveness for limbs, as I've showed you before. But you can see that there are increases in distinctiveness of patterns that we didn't actually show increases in the volume. If, for example, there's an increase in distinctiveness for numbers and also increases in the distinctiveness for houses in the right hemisphere. And there are not significant changes in distinctiveness for other categories. However, if we take the non-selective voxels of lateral VTC, there are actually no significant changes in distinctiveness across development. So there are kind of two findings here. One finding here is that the changes in the patterns are really driven by changes in the selective voxels. And second of all, we have this differential development of distinctiveness across different categories. So to better understand how that affects category representations, what we did is again used MDS to look at the representational space in children compared to teens, but this time focusing on the selective voxels of lateral VTC. So this is the representational space of 5 to 9-year-olds. And again, you can see that the faces and the body parts seem to be separated from the other categories, but the numbers and words really are not separated from the places. But with these changes in distinctiveness, what we see is an enhancement of category representations. And in particular, you can see that the word and number representations become much more separated than the other categories. And you can see even some changes in the face categories that in the young children, the two kinds of faces were overlapping, but there's a little bit of distinction now in the patterns from child faces and adult faces. If you look at the non-selective voxels of lateral VTC, you can see that there really isn't any clear category information in these voxels. And in addition, we see no developmental change. So this is kind of nice. We can see how the representational space of children changes across development, but does it have any impact on perception? So one possibility is that the developmental increases in category distinctiveness, especially for faces and word, might be coupled with better recognition performance for that category. So to test this hypothesis, we wanted to first assess the relationship between performance on a task, for example, reading, and distinctiveness for that category, for example, words. And we want to see how predictive this is. So what we do is we measure this relationship in N-1 participants, and we use an leave one out cross-validation approach to predict, given the model parameters from the distinctiveness pattern of a left-out subject, how well they're going to be able to read words. And of course, we're going to do this prediction across all N folds. So this is to give you a sense of how the relationship looks like. Again, the x-axis is a distinctiveness for words. The y-axis is a reading performance. And you can see that there is this positive relationship where higher distinctiveness is coupled with better recognition performance. From this relationship, we extract the model parameters or the slope, and then we take the distinctiveness of the left out subject and predict how well they can read. And our prediction error is pretty good. So we can predict the reading performance up to with an 8% error. And if we add other factors like to the model, like TSNR or age, for example, this is the age, we don't really improve the prediction. So it suggests that this distinctiveness can predict reading performance. We did a similar analysis relating distinctiveness for faces compared to face recognition performance. And again, there is this positive relationship with more distinctive responses is coupled with better face recognition. And when we do this leave one out cross validation, we can predict face recognition performance with about an 11% error. So just to kind of summarize the second part shows that there are both increases and decreases in the distinctiveness of distributed responses during childhood. And this overall enhances category representations in lateral BTC. And also that these increases in distinctiveness, especially for faces and words, are coupled with better recognition performance for that category. And just to end, I just want to make a kind of one more point about the relationship between this functional development that we can see in our children to anatomical development. And the reason that we're interested in this relationship between functional development and anatomical development is that changes of functions might be coupled with changes in anatomy. So one possibility is that these functional changes in representation or selectivity might be associated with changes to connections to a neuron, and particularly some researchers like Carla Schatz or Pascal Rakic have suggested that there's pruning of irrelevant connections, and this pruning of irrelevant connections might lead to increases in selectivity. Alternatively, changes in selectivity might be coupled with changes in dendritic arborization and myelination, because there's a lot of other evidence that suggests that myelin is activity dependent, and as we become more proficient in using a piece of cortex, it might produce changes in myelination. So it's really hard to measure myelination or pruning in vivo, but we can use new methods like a quantitative MRI that has been developed by Aviv Metzer and colleagues to look at changes in tissue density. In particular, with quantitative MRI, we can measure the T1 relaxation time of tissue. And as tissue becomes more dense or the macromolecule tissue volume increases, T1 decreases. So we can track if there are developmental changes in T1 time that's associated with cortex either becoming dense, more dense, or less dense. So previously, we've shown that there is this developmental decrease in T1 relaxation time. So tissue becomes more dense or it grows. And interestingly, this reduction in tissue relaxation time is associated was increased functional selectivity. So there's this intimate coupling between increased functional selectivity, for example, in the face area, and the fact that the tissue becomes more tissue dense. Now, as Sharon alluded in the beginning, we're really interested to understand what parts of the tissue are making it more dense. So one of the things that we're interested in is in myelination. And we have now accumulating evidence that this might be the case. So one way that we did that is looking at ex vivo tissue samples. And we compared the tissue relaxation properties of ex vivo samples with quantitative MRI, shown here, for example, in a sample on the right, and with myelin staining, shown here in the left. And basically, we cannot do, of course, longitudinal measurements in postmortem brains, but what we could do is we can look at regions that have different T1 properties, like the fusiform gyrus versus the collateral sulcus, see their quantitative T1 properties with MRI, and compare that to actual myelin staining of the same tissue sample shown here on the left. And just by eyeballing, and you can look at all the quantitative analysis in our paper, you can see that as the tissue is darker, meaning it's more tissue dense, you can see that in the T1 image, this is a high resolution, it's 100 micron. And if you can see in the same sample, you can see that there's quite a lot of myelin in the cortex. And in fact, if you look at the fusiform gyrus, myelin extends up to about half the cortical's depth. So we think that this contrast that we're seeing with MRI is really associated, even in cortex, with myelination. So this suggests that this decrease that we see from childhood, which is to adulthood in the T1 relaxation time, is probably not associated with pruning, but is associated with tissue growth. But we were interested to see that maybe pruning happens earlier in development. So more recently, we've extended our quantitative MRI to look at tissue development in young infants. So we've scanned 10 young infants longitudinally over a period of 6 months. So at newborn, at 3 months, and at 6 months. And we've looked at the T1 properties across all of visual cortex. Here I'm showing you data from ventral, from like all the retinotopic regions of the ventral stream. And you can see that in the young infants from 0 to 180 days, which is 6 months, you can see these really big decreases in T1 that are again associated with tissue growth. And to get another view about what might be driving this tissue growth in infants, we've used some genetic data from the Allen Institute and compared gene expression in postnatal samples. So these are 4 months old babies compared to prenatal samples. And we looked at them in visual cortex separately, but also across other primary sensory areas. So you can see that there are some genes that are much more expressed postnatally than prenatally, and these are indicated by the pink and red colors. And it's really interesting that these genes that are expressed prominently after birth, of the top 10 genes, 3 of them, MBP, MOBP, and MAG, are associated with myelination. So I will summarize here. So I have 3 take-home messages for you today. One, I've shown you evidence that development of cortical function doesn't always follow a monotonically increasing trajectory, that we see these developmental increases in distinctiveness and selectivity to faces and words, and they're associated with better face and word recognition abilities. And as we see these functional changes in cortex, we also see anatomical changes. And we believe from accumulating data that myelin is a key contributor to increases in cortical tissue density in the visual system during infancy and childhood. And finally, I would like to kind of end with a thought about cortical recycling, kind of zooming out a little bit. So the question is, why do we have cortical recycling during childhood development in the first place? So we hypothesize that cortical recycling during childhood development might reflect an adjustment to visual demands and frequency of visual stimuli that are important. For example, young infants might look a lot at faces, but toddlers look more at hands as they learn to manipulate objects. And later in childhood, other visual stimuli and tasks, such as words and reading, might become more frequent and important, which might lead to recycling of prior cortical representation for the more salient and behaviorally relevant stimuli. And future research that can measure the visual diet of what children look at, together with their brain development, can shed new light and test this hypothesis. So thank you for your attention, and I'm happy to take any questions right now.
Thank you, Kalanit, for a really lovely, amazing talk. And I'd like to ask everybody to unmute yourselves, and let's give Kalanit a big thank you for a wonderful and many new exciting results that you presented. So of course, I'm opening the stage for questions. I myself I will start. I don't tend to do that, but I want to ask about all these voxels that you found that were non-selective. And I want to ask if you think that they are non-selective relative to the categories that you have tested, or do you think that they're non-selective in general?
Well, I can only tell you that they're non-selective to the categories that I've tested. And of course, if you think about the number of nouns in the word in English, there are 30,000. So there's quite a possibility that they might be selective to other stimuli. People have looked at this and they haven't really found a prominent category selectivity to other stimuli. There's a new data set by Kendrick K. It's called the Natural Stimulus Data Set. It has 10,000 images. So if somebody wants to look and find new category selective regions, go for it.
Okay, wonderful. There are questions in the chat, but if people want to ask questions now, well, we can start with that and then I'll read them. Or you can ask them yourself, Dani, Eliran. Okay. So I'll start with, Noga Vardi says, fantastic talk. Besides myelination, there must be an increase in synapses. Okay, I don't know if that's a note or a question.
So I can comment on that. So when we did the transcriptomic gene analysis, you can do an analysis. There's a toolbox called Top Gene, and you could see what physiological processes these genes are involved. So we put all the top 100 genes, and there is definitely, they're involved in other processes. One of them is synapses, you're absolutely right. Another thing is axonal and dendritic arborization. So I'm sure these additional things happen. We can't measure them with MRI. I'm not sure if so the question is whether we can measure them with MRI or not. With myelin, there's been a lot of research outside my lab that shows that these contrasts are sensitive to myelin. So this is why I say myelin is a key contributor. I didn't say it's the only contributor. What we're doing right now in my lab, we're collaborating with an anatomist at UCSF. Her name is Mercedes Paredes, and we're actually doing immunohistology staining for multiple tissue compartments to see actually what is changing in this cortex during development, because we might be blind to things with our in vivo measurements.
Okay, thank you. I see that Ella has asked precisely the same question as I've raised. So, and she also says, thanks for a fascinating talk. Dani Harari has asked a long question. Dani, do you want to ask yourself, or do you want me to read this?
Sure. So this is also related to your question, Ella's questions, because I was wondering about social understanding, which is developing significantly in these ages, mainly after the age of 5. You may say that this social understanding in general is related to faces, limbs, body, and maybe even some word categories. And I was wondering if you think that this understanding, the development and the representations are actually competing or racing with the other categories you mentioned, or maybe they are developed in these non-selective areas that you found.
So I'm really glad you guys are asking these questions. I think the limb recycling has to do with social and communicative development, because I think one example about why children look at their hands is because they have to look at things in order to manipulate them. It really helps them. But it's also about communicative, right? There's a lot of pointing and gesturing. When you're a toddler, it becomes very rude to do it after a certain age, right? So there's much more nuance in the facial expression, in the eye movement, in the gaze. And I think children, it becomes a better communicative tool, and also it's more socially acceptable, right? So I think you become fine-tuned not only to the statistics of your visual environment, because of course you see faces from the moment you're born, but what kind of information you retrieve for the faces. This is one thing. The reason that I think that the face selective regions are developing is not only because of this, is because also your social network of people really increases a lot across your development. So if you're a young child, you have your like small circle of family and maybe your kindergarten class, but by the time you go to high school, your high school might have 500 or maybe 1,000 kids, and they all kind of like your age. Their aspect ratio of the faces is similar, right? So you have to become much more fine-tuned to a larger cohort of people. And of course, you have to continue to learn faces throughout your lifespan, right? So I think this is what's putting pressure on the face area and why it's still increasing all the way into teenage years, which is actually quite a long time.
Yeah, so Joseph raised a hand. Joseph, do you want to ask?
Yeah, for sure. Great talk. Thank you. You've raised a lot of questions in my head, and I'm going to ask just a few of them. The most important one I think is important for our work is, how do you, in the children, as they're developing, do you keep track of each individual person's brain getting larger?
Okay, so we measured brain size in our, when we started doing the cross-sectional study, we were really worried that the kids have smaller brains than adults. It turns out that after age 7, it's not the brain that's changing so much, the size of the skull is changing, and especially males develop a really thick skull. Really, sorry, it is what it is.
That's good that they're the ones who play American football then.
Yeah. So basically, in children, it's not an issue. When we look at the myelination, we were looking at cortical thickness, because there's a lot of data that shows that cortical thins across development. And we think that it's actually not thinning, it's getting more myelinated. And because myelin increases the contrast, it makes the brain looks wider, it changes the boundary of the white and gray matter. So, and we actually looked in that study, if you look at Vaidi's PNS paper, we look at the relationship between area changes, thickness changes, and myelin, or it changes in the T1. And it really looks like that it's a myelin in the fusiform better predicts this cortical thinning. With the babies are really fascinating. It's a whole different story. Yeah. Their brains are contrast inverted. Their white matter is darker than the gray matter. All the tools that we had to use, that we can use, we can't use them. So we have to use new tools for the babies. The babies have a profound change in size. So what we haven't done is look at the connections yet, but we can measure what we do. We can measure the same area over time, and that area, independently of the size changes, it is changing its density basically. But you can physically see the baby's brain growing. I mean, it definitely does, right?
Yeah, you could just measure their head and you would see that. Yeah.
Well, we're going to measure the brain, but their brain is
Yeah.
it's changing. So what we're going to look at is like areal changing and also the deepening. So the babies are born with all the sulci and gyri. We've looked at that, but the sulcus and gyri become kind of deeper, and the gyration index changes. So we're interested in looking at that.
Yeah, and so I have like so many questions. I could probably occupy the rest of your life because we've talked about this stuff before. So one of the things I want to know is when you measure the density using T1, is it a typical normal T1 scan or is it a different one?
So it's
normal one?
No, normal one is called a T1 weighted scan. It doesn't give you units. A quantitative T1 scan is a scan where you do multiple flip angles.
Flip angles, okay.
Yeah, and multiple SPGRs. And basically, you know, the T1 is a decay, right? So in order to fit the exponential decay, you have to have multiple measurements.
More points.
Yeah. So you have to do a special scan. The high resolution scan takes between 20 to 40 minutes. So,
all right.
Yeah. The babies we scan when they're asleep. So that's how they don't move for such a long time.
Yeah, and then like, so then I was thinking about looking at my longitudinal data in people with Parkinson's using your tool, but I already collected the data and I used a normal T1, so I can't do that. That's unfortunate. I was thinking about using this as a measure of plasticity, but in reverse, looking at neurodegeneration. But from what you just told me, in our studies, we have to apply a new scan to each individual person. Okay, we'll do that. We'll put that in the grant. Okay, and then the last question, I'm really intrigued with this hand area. I'm just gonna say a quick one. Have you looked in the hand area? Have you looked in the hand area in S1 or M1, or do you only scan the ventral occipital?
So we've scanned the whole brain, but the hand area does not respond strongly to still images of hands. So we don't really see a whole lot of selectivity, but we haven't targeted it specifically as an ROI.
Okay. That's great. Thanks.
Thank you. Ella?
Thanks again for the great talk. I have a question that kind of follows up on the previous discussion of how kids need to be attuned to more different faces and acquire a broader, distinctivity between stimuli. And I was wondering whether you've had a chance to look at within category sensitivity and selectivity throughout development, which is slightly more attuned and slightly more in line with the behavioral testing of face perception and so forth. And a related, but not exactly related question, I'll just throw it out there and maybe we can talk about it some other time, is when you're testing for limbs, what limbs are you testing for? And do you see a change in balance between hands and legs? If both representations decline or mostly the legs, I don't know what you did over there, but you know.
So our limb category had an assortment of hands, arms, feet, and legs. So we didn't separate them, but probably we should in the future studies. So I can't answer that question. With regards to within category sensitivity, we did that in a cross-sectional study. We have a 2016 paper in Journal of Neuroscience. Vaidi Natu is the first author on this, and we use an adaptation to measure sensitivity to changes in face identity. We did it separately for child faces and adult faces, and we see that there is basically a faster release from adaptation. So we varied the morph level, basically. We varied the similarity among faces. And basically, adults have a steeper slope, so they're more sensitive to changes in face identity. And we have a little bit of an other age effect. So the children are a little bit better on children faces, and the adults are a little bit better for the adult faces. But overall, adults are just better than children. And it is coupled with perceptual distinction or perceptual dissimilarity measurements in the same participants. So we did that for faces. For words, it kind of makes sense to really look at reading. So we use a variety of reading tasks, basically. It doesn't really matter which reading task, or it doesn't really matter which face task. It seems it's very similar.
Excellent, thank you.
Okay, wonderful. There's in the chat, there is a question by Jacek, or I'm not sure I'm reading this properly. Jacek, okay, great talk. Could you comment a bit about the distinct roles of myelin in the cortex in comparisons to its role in the white matter? As I understand it, based on recent review papers, myelin within the cortex has a regulatory role and is actually, and it actually prevents creation of the redundant dendritic connections on paper. As a mechanism, this sounds more like live pruning than recycling. Could you comment on that in the context of the functional results?
So I'm interested, maybe you can write in the chat which paper specifically you're referring to, because I'd like to know about it. So that would be useful to me. I think there's been a lot of interest in the last decade or so, even more. I think it's since Heidi Johnson's work, how do you call it, juggling study. She started with VBM, that kind of learning changes the anatomical properties of tissue. So the kind of things that I know about is that generation of myelin is activity dependent, and specifically it's regulated by oligodendrocytes. So there's our oligodendrocytes in cortex, and basically activity of neurons can generate these precursor cells to become oligodendrocytes, and they generate myelin. I'm not sure about how, so basically you're suggesting that the axons that get myelinated are the ones that are going to remain and the other ones could get proof. So that could be one mechanism. Another possibility is that it basically increases the efficiency of transmission. It makes it less noisy and also might improve kind of the neural function. This is kind of like my thinking about this right now.
Okay.
Actually, what you say?
No, I'm just saying that he still hasn't written the names of the reviews, but maybe he will send it by email to you or to me, and so I'll forward it to you. I mean, I'm not familiar with these.
Okay. I have a question for you guys, actually. So a lot of you guys here studies the ventral stream. It's very clear to me what is a task we need to do for faces. It's very clear to me what is a behavioral task we should do for words. It's really less clear to me what is a task we should do with limbs, because nobody, maybe I'm a limb agnostic, but I don't really think that people go around the world recognizing people or limbs, right? So I'm really thinking about what is the right task that we should add to our studies to look about the function of this limb selectivity in ventral temporal cortex. Is it about gesturing? Is it about, I need some help here. Some ideas.
So I really want to ask, because you showed, okay, this is a sort of a reply, but not a full answer. So you showed us results from the ventral aspect of the cortex, but that does not, if I understand correctly, include what is known to be EBA. And I wanted to, I mean, you probably have the EBA related data, and I wonder if there's a trade-off between like these ventral areas shrinking and EBA growing, or some.
EBA is huge in little kids. It's just huge.
Also in adults, it's quite, but it shrinks in adulthood, or it,
I haven't quantified it, but I'm pretty sure it's going to go in a similar direction.
And do you have, I'm wondering if you have any diffusion data or tractography that you can look at the connectivity. Okay, of course. That you can look at, maybe the connectivity also changes with age. I don't know.
Yeah, so we have a bioarchive preprint. Right now it's under review. We're looking at the actually the word and the connections to the face and word areas across development. So if you're interested, you can look that up. It's Emily Kabuda is the first author on that one.
Okay, definitely. Okay, so I see Ella wrote something. I mean, Ella, do you want to say it yourself or shall I read it?
I can say it. I just didn't want to jump up and interrupt everyone. Maybe, again, from a dorsal perspective, and I can't imagine a ventral perspective that would be separated for that for limbs. Things like action and gesture identification. I'm sure there are point light display versions of this, and this is what we kind of use in order to identify things that are more dorsal. And again, you can do a version that's real hands versus point light displays. And point light displays just tend to be harder to identify fine-tuned distinctions. So it's a more sensitive task.
But what would be the task? Tell me the action?
Tell me the action. Yeah, or even discern which to act, if it's the same or different action, if they're very similar.
Okay.
That was also my intuition. I would also go just with the name of the action that is being carried out. It was like the most natural thing to do with the limb.
Did anybody have like a database of like videos with action that are agreed upon or rated upon? I guess we would need to develop that.
I think I know something similar for whole body movements, but I don't know one for limbs per se.
Yeah.
And I think, yeah, that may need some
Yeah,
development.
whole body don't develop, right? So that's not going to be.
Kalanit, what portion of your limb stimuli were hands?
About half and half.
And okay, because I was thinking maybe you could, I mean, maybe the shrinking part is the not hand part. I don't know.
That's what I was also thinking. From studying people born without hands who do things with their feet, this distinction between responses to hands and feet would be very curious.
Yeah. So Katrina suggested to look at sign language, right?
Yeah.
Yeah. So
Yeah.
Marissa's going to do that. So she's really interested in studying deaf children, because deaf children would generate a prediction that their hand limb representation shouldn't shrink, because it does have social salience and communicative salience throughout development. So she's going to take that route. I'm still trying to think about what to do with just typically developing children.
Sharon Gilad Gutnik wrote, okay, I thought maybe you want, how about recognition of specific gestures? Sharon, do you want to elaborate?
I mean, I don't have like a specific task in mind, but if you're looking for still images and you're looking for things that have probably pretty clear boundaries already for children, then like pointing versus, saying hi, you just wouldn't have a big range of gestures probably to include, but those might work.
Yeah.
There's another question by Eliran. He's asking if you think the recycling and specialization also happens in somatosensory and motor cortex, or do you think this is specific to ventral sensory cortex?
So what would you predict for recycling in somatosensory cortex? So you would expect that because you guys asked me about the hand region, the representation in motor cortex would decrease over development.
Maybe
Yeah.
the hand, I don't know, maybe the hand would increase, but I don't know what it would
But isn't all this research like in violin? I think like in violin players that you become more like the one hand has, I'm not playing violin, obviously. One hand does all the work and the other one just goes like this, and that hand gets expanded. So I guess, I don't know what's competing there basically. So I don't know why that might happen. I think there has to be some pressure, right? And some changing tasks across development. So reading is kind of interesting because you clearly learn it later in life. So maybe if you want to look at hand representation, it would be interesting to look at people who learn to play an instrument or a skill, maybe athletes or dancers that learn a skill later on in life rather than earlier in development. Yeah. For me, it's the most surprising thing, to be honest, in this research is that we find these decreases, because coming into this research, I was sure that everything is like a refinement, and I was shocked to see that there's also some losses. Or another way to think about it, children are not mini adults. They're actually better at us at some stuff. So.
Yeah, and the limbs, they come out of nowhere. That's also surprising. Like nobody has put them up as in any hypothesis. They were just, came from the back door. Dani has said, thank you for a very interesting talk. And also there was another one like this. Yes, and Katrina also said, I have to go. Thank you for a super interesting talk. Michal, also, I have to leave. Michal Ramot, thank you, Kalanit, for a great talk. And Ella, again, you have.
Sorry. This is a comment based on a comment. Don't just.
Okay, fine. I'll send it to Kalanit later. Sharon, I see that you've got a question.
Yeah, I mean, this actually, I think, maybe relates a little bit to what Ella just mentioned, and it's not really well formed, but I'm just wondering, in terms of all the categories, if you have any predictions about, especially in the context of like word recognition and the fact that reading and writing comes in later, what about like basic symbol recognitions or of drawings that, stand for certain meanings, because children begin to draw and kind of fine tune those representations, and those lead or are very related to the development of their writing skills, but they come on a little bit earlier. Do you have any thoughts about that?
Yeah, so I think my thoughts are that it has to be a visually important task. It has to be a task that involves your foveal resources, and it has to be a task that is really meaningful for you socially or personally, and you spend a lot of hours doing it. So the examples that I have is from studies that I did with Jesse Gomez, is a Pokemon study, right? So if you have kids that spend a lot of time, hours a day, a week, looking at Pokemons, they will develop selectivity to Pokemons. But the Pokemons, at least in the mid-90s, were kind of like pixelated and they're pretty similar. So you really had to use your foveal resources. So I think that's something that is going to put a pressure on this. It is the case that these medial VTC regions that are more peripheral seem to be developing earlier. So I'm really intrigued by the fact that these foveal areas seem to have much more prolonged development than these medial areas that deal with just in the periphery, basically.
But what about things like, basic geometric shapes and symbols, which have a value for communication and socialization, and that they probably kind of deal a lot with because they're scribbling, and then it turns into more like recognizable shapes, and then.
Yeah, I think it has to become automatic at some point, right? At some point when you read words, you don't do it letter by letter, you read the whole, right? You have this holistic processing. So maybe if we did the street signs, there are definitely other symbols that this can happen, but it's not general text. This is why we did the numbers, right? Okay, I don't want to keep you guys all here forever, so I think it's a good time to end. It was really great seeing you.
Thank you very much, Kalanit, for a wonderful talk, and a few more things I'll send you from the chat so you can have a look at additional comments.
Okay.
Thank you again.
Thank you for all the questions.
Thank you again
for
for joining us. It was a real pleasure.
Bye.